Hello, third year student from Auckland University. Here's my talk to you about what I do, what I'm interested in. And it's really about the application of basic science for plant defense engineering. But before I dive in straight in, just a brief background where I come from. So I come from the German countryside and from a baking family, as you can see here. So that's like the bread which my brother was baking. So when he's leaving the shop in the morning, there's a lot of bread. So I have a huge affiliation with food and the production of food. And in contrast to my brother, I don't really want to get up every day at 1 o'clock and go to the bakery and leave at 8 or 9 a.m. in the morning. So I thought it's better to do some studying and go for plant science. So together with wheat, rice is really the major crop. Together, they produce roughly 40% of all calories consumed daily. So they're clearly a rich source of energy. Not only us, but also like microbial pathogens and interested to harness this energy and grow on it. So, for example, viruses or mycetes, fungi, bacteria, so all phyla of life actually attack plants. So we are in direct competition with plants, and I'm going to talk to you about how a bacterial pathogen actually infects rice. And as you can see here, that's a picture of an infected and unhealthy rice field in India from our collaborator. In the back, you see a rice variety which is different in one single gene from the one in the front. It's this resistant rice variety which is immune against infection. It's totally healthy, and you can imagine you can harvest a lot of grain and feed a lot of people. But at the front, this bacterial pathogen infected these plants. These plants are like immune compromised, so to speak, and they're totally susceptible, and you're not going to harvest much grain from this. And that's not really what you want. And so that's what really my interest is how plants defend themselves. And in a cartoon structure, this is rice, and rice gets infected by the bacteria, they enter the leaves, they start to multiply, and then you can imagine two contrasting outcomes, as shown in the picture before. One is susceptible, that the rice doesn't recognize the bacteria, it's just like blinded, the bacteria are really happy walking, multiplying and growing inside the plant, using up all the energy, and the plant wilts and dies, as shown in the foreground with the yellow leaves on the previous slide. And you imagine there are also like plants which are resistant, and these are green. In this case, the bacteria have like antennas and like sensors which can recognize the bacteria, and this recognition leads to a defense response, and then the plant can fight off the bacteria and stop multiplication. And so in a cartoon form, this looks like following in the plasma membrane. We have like receptor like kinase mostly, which recognized molecules from the bacteria, or in other cases fungi or mycetes. And together with co-receptors, this leads to signaling and a disease-resistant response. But also pathogens are able to secrete molecules inside the plant cell, and these are often called effectors, and these can be recognized by different class of immune receptors. So the take-home message here is there's really like two different layers, if you like, from the immune system in plants. One is happening at the plasma membrane, and one is happening intracellularly. And I'm studying mostly the immune system at the plasma membrane. So, and how do you measure this? How do you actually study this? So when these receptors get activated by a molecule from the bacteria, in our case, you get a plethora of defense responses like reactive oxygen production, calcium, and kinase. And you can really measure this. And as I said, we were interested in looking at the interaction a bacterial pathogen, Xanthomonas, in the rice. And in rice, we are studying this immune receptor XA21. And this is how it looks like in real. Then, if you have a rice variety which outs the receptor, you're blind and nothing happens. But if you actually have the immune receptor and the right molecule from the bacteria, which is recognized in our case was called RUX-X, you see like a nice immune response. And this is just a measure of the reactive oxygen production, which you can see here is happening within 1 or 2 hours. Then you can also measure by quantitative PCR production of certain defense genes. Here is like pathogen-related gene 10B. And again, if you don't have a receptor, you're totally blind. If you have the receptor XA21, then you can actually sense this RUX-X molecule and you have a defense response. And the last step we are really interested in is really like the disease outcome, so the additive effect of all these responses. And as you can see here, if a bacteria is recognized, it doesn't really grow much, so your leaf is actually green, and you stop a bacterial progression. But when you take away this gene in the bacteria, the plant can't recognize the bacteria anymore, and the bacteria can happily multiply. And this you can just simply measure. So short lesions, you're resistant. These are lesions. So if they're short, then you're resistant. If you take away the gene, which is recognized, then you become susceptible. So you get this long lesion. So you just measure this. And if you put back the gene to complement the mutant genetically, to show that only this gene is really responsible for this, you become resistant again, so you can recognize this bacteria. So what I just briefly showed you, which is like a lot of work, and I always encourage my students to actually go read the primary literature, and then you come back and we can discuss it, is that if you have a rice plant which has this immune receptor XA21, it recognizes the molecule from the bacteria which is called RUX-X. This is a molecular recognition event. This leads to immune response. On the other side, if you lack this receptor, or you don't have a RUX-X present, there's no immune recognition, and you become susceptible. And so in future, what I'm interested in to apply like similar approaches to stop like kiwi fruit pathogens, for example. So kiwi fruit, as a major crop in New Zealand, and there's like a bacterial pathogen will infect this. And what we want is not that the pathogen can actually feed on the tree, but really that we can harvest like a nice crop and produce kiwi fruits in the future too. Thank you very much.